Okay, let's start the class, and we are gonna do the second lab. Second lab, we are moved to, we say smear preparation and simple stain. And this is going to be the first time you guys really touch the bacteria. We'll try to make it simple, so you will have is E. coli. Now, by the way, this is what we talk about in the lecture. This is the agar, which is created by Fanny Hesse in Rob Koch's group back in 1800s. Used to grow bacteria. It's polysaccharides, extracted from the polysaccharides. And the container is called a Petri dish, created by Richard Petri, and it is a round shape. It's a plastic one. And this is disposable. When we finish, we just autoclave it. And you see the bacteria is streak there, but this is not a way to do streak plating. This is only to grow the bacteria. So the purpose today is we pick the smear from there, we make the sample, and we are going to do the simple stain. Okay, that's the purpose. But it's not really the way we're supposed to do the streak plating. Just want to let you know. So first of all, why we can do simple stain? Bacteria on the plates like this, or bacteria in broth. So let's say we have agar plates. Okay, we are growing bacteria. You have a broth, 37 degrees Celsius, 24 hours. The bacterial cell culture, let's say like this, is negatively charged. And then all those die. Where are the die? It's basically on the side of your box. So you can see you have a crystal violet, a purple color. You see a saffroning, a pink color. You see a methylene blue, like that. Or sometimes you have a malachite green or carbon fusion. Those are we call the chemical dye. Those dye are positively charged. And the negative and the positive can be stacked together. So this is like a chemical dye. And this is a bacterial cell. That's why they can combine together and do the staining. So it's a very simple principle behind it. Now the next question. What are the chemical dye we're going to use and we can use? There are several we have in the lab, not too many. And you can pick any from them. The first one is very common and looks very purple. Crystal violet. Okay, if you cannot see it, crystal violet. The second one looks very pink. Look like that. Or too red. Saffrony. The third one, it looks like blue. So we call it methylene blue. And sometimes later on, we have another dye, which looks very green. So we call it malachite green. And also I want to say today what we're going to do is we are staining the bacteria, not staining the background. So this simple stain said very easy. It is positive stain. There is another different method of the staining. It's called a negative stain. We stain in the background, and the bacteria is showing up. Okay, it's a very basic knowledge. So how are we going to do it? It's a step called the smear preparation. Okay, first step. You will get a glass slides. A glass slides will be like this. You can pick from the front desk, or if you have in your personal tray in the box, you can got it like this. It's pretty clean. So first thing what you do is draw a big circle. Today we only going to use one string, is E. coli. So on the corner, label E.C. which means E. coli. The acronym of E. coli. Okay, then the second one. This has to flip over, because the Sharpie marker, especially sometimes people using crayon or those type of chemical marker, could be damage the lens. So the glass slides needs to be flipped over. Okay, if you flip over, pretty much like what this means, the CE like this. Okay. Okay, so flip it over. Then what we do, will follow next step. We will add a drop of water. But before we add a drop of the water, the loop has to be sterilized. I'll show you how to do. Okay, you have a loop like this. So make sure it is straight. And we'll just turn on the tap water. But before we do, make sure this is you flamed on the Bunsen burner, and I'll show you how to turn it on. And then add a little bit drop of the water. Okay, right in the middle. So this will be flaming the loop. Add one drop of water. The next step, once this is there, okay, let's say we have glass slides. You have circle, you have a label, you have a drop of water. We're going to use a loop to pick, I will say that not the colonies, the bacteria smear from agar plates and make it's a smear averagely covered on the surface of the circle. Make sure this smear is thin. This is important. We don't want a very thick one, because we would love to see the morphology and also the arrangement of the bacteria. So make sure the smear is thin. Okay, you have water, you have a bacterial smear. We're going to move to the next step. Air dry for 3 to 5 minutes. This is important. We want to be completely air dry, which means the dye and also the bacteria could be kind of stick on the surface. Next one is called heat fixing. Heat fixing, if you have a Bunsen burner like this, okay, you have a fire, basically a glass slide is on the surface. We're going to do a back and forth heating to make sure the enzyme has been cured. The bacterial enzyme has been cured and firmly stick on the surface. That's called heat fixing. But remember, it's not cooking. Okay, so very gentle back and forth. Something I want to mention, we need to do the air dry first, then do the heat fixing. If we do not finish the completely air dry, directly move to heat fixing, what happened? The agar plates, the glass slides later on underneath microscope, you will see lots of things like this. Lots of legs will come out. If it's not completely air dry, directly to do the heat fixing. So air dry and heat fixing are two steps are very important. The next one, we're going to add 1 drop of dyes and staining 60 seconds. So any of the dye from here you can pick. Some of you can do crystal violet. If you are doing crystal violet, then your bench partner can do saffron. Then we have a different color, we can do the comparison. If somebody using, I think we have crystal violet, saffron and the malachite green. Some people using methylene blue, then the other people can do saffron. Then we have a different color, we can do the comparison. So add 1 drop of the dye, staining for 60 seconds. Then a gentle rinse, about like 10 seconds. Okay, using tap water. Not flushing, very gentle. And then we dry. Okay, dry a little bit. Then what we will do, this glass slide is finished. We will using 10x to find specimen, and then we move to 100x with oil immersion. You are going to see the 2 important information is morphology. This means the cell shape. And we're also going to see the arrangement. Okay, what is the morphology? Morphology encoding something like this, dots. We call it a coxel. Sometimes it's like a line. We call it bacilla, or we call rods. Later on, when you see when we learn the passaging of the bacteria, lots of them, like that, like this, like this. It's like a coxel, but like a rods. So we say Coccobacillus. And you will also something like this. That's called a vibro. That's a morphology. Okay, what are the arrangements we're going to talk about? Arrangement is very important, especially when we move to the exam 2, we talk about all those passages. The arrangement is very typical for several human passages. For example, it could be the two coaxials stacked together. Okay, that's called dipole coaxial. It could be just a single. So we say single rods. It could be four of them stacked together. We call it a tetrad. Okay, that's some several examples. I give that to you. Arrangement also could be a very long chain. Okay, like that, connected together. It's a streptococcus. It could be a long chain. Okay, those are the arrangements. This thing we can use in simple state to do. The missing one is the gram results. That will be learning on next week, Tuesday. Okay, so here are some of the examples. Then I'm gonna go ahead to show you how to do. It is the first time we'll touch the bacteria. So like I said, you're gonna wash your hands first. And after that, you wear the gloves. I'll move to the bench right here. Then everybody coming, and I can show you how to do it. Okay. So the camera will be moving around this area. So I can record it and show you clearly what we're gonna do. Okay, you guys are gonna be there in the YouTube channel. Okay, so what we're gonna have? I will borrow this. Okay, we'll have a glass lights. Will we have a, let's say, rack or a tray, a faucet, a wood faucet, and also a Sharpie marker. That's what you need. And all these guys. Let's say today I'm gonna using saffroning. How about? And then we're gonna pick the bacteria. So the bacteria using E. coli will be right here. Okay. These things are kind of like here. Now we want to do one thing first. Is this the first time we're gonna touch the Bunsen burner? So I want to show you how to use it. Bunsen burner, this will go. Do you mind a little bit here? Okay. Bunsen burner seems like you guys are shared. This was put on the cell phone, put on the side. Turn it on. You should hear kind of the voice. That's tricky sometimes. See the voice? That's an air. Sorry, changing mode. Doesn't work? Can I have a new one? Oh, well, just give me the hose. Give me the hose. The hose. That works. Okay. Then this one works. That means this one also works. Otherwise, I'll. Okay. Never mind. We'll put it right here. So can I have the?
I think you're going to do it here.
I'll just do it. Take it off. Okay. So that's tricky. Here we go this way. Look at that. Looks like a locket. That's how the fire. So that's a fire. Okay. At the bottom, you can adjust it. Huge and small. You don't need too huge. The second one, what we want to check is this water is working. Sometimes the water doesn't work. Okay. So at least it's dripping. Okay, that's too much. A little bit is fine as long as it's dripping. So we want to have a loop ready. Make sure these things are ready, okay? Straight. You need to use a loop, which means on the top there is a circle. This is not a loop. That's called a needle. There's no circle. We will use it later on when we do the biochemistry test. So these are the things, just make sure it's ready. Okay, now what we do, label. Draw a big circle. Right on the corner EC. And then flip it over. Okay, now using loop. Make sure it's sterilized. Flame. It has to do like this, 45-degree angle, little bit on the top. This is not correct. Some people like this. No, that's not correct. The whole thing. Okay. Now what we do? A little bit drip. So how much water is good enough? Okay. Flame it again. It's very important. Now, touch bacteria on here. Little bit touch on the surface of the agar. Cool down, then you pick some bacteria. Let's say you can pick anyone. Okay, I'll pick some. Maybe 2 colonies. Okay, now make a big smear. Average on the surface. Average on the surface. Okay, flame it again. Now you're going to wait for 3 to 5 minutes to do the air dry. Let's just say 3 to 5 minutes already. Back and forth, couple of times. This is called heat fixing, but not cooking. Okay, you have to very average to do. Make sure don't burn yourself. Okay, back couple of times. So it's ready. Cool down a little bit. It's very hot. Don't touch it right now. That's why it's important that you need to have a gloves ready. Okay, now go here. Little bit close. Put right here. Okay, a very important steps we need to emphasize. At this moment, you need to wear goggle because it is important to protect your eyes. All these dye, if that's somehow touch your eyes, the federal regulation is using that flush water is to splash your eyes for 15 minutes. And it never happened in my lifetime, teach like 12 years. 2 years in other school and 10 years here. But you still have to be cautious. Wear a goggle. The goggle right in the middle of the box. Or you can bring your own, okay? Now we're going to go ahead to do the stain. So I use saffroning. A big job in the center. That's too much on this thing. That's a little too much. So wait to dry about staining. We call it a stain. Okay, staining about 60 seconds. Let's say 60 seconds is ready. Then this is, make sure it is working. Okay, then you have your faucets right here. Very gentle. Okay, now finished. That's all. This guy, because it's touched the dye, it count as biohazard trash can. So make sure it's right here. If that's not a general trash, okay, very important. Everybody should know all these dye, it's count as biochemical hazard. So then, let's do one more time to show you how to use microscope. So I want to make sure this is working. I'll borrow this microscope as an example. I hope it works. Maybe it doesn't work. Okay, we'll see. So how we do it, you should be refresh what we did on the first lab. Okay, how we do it. Turn it on. Somebody said 100 lens damage. I don't know. What's damaged?
This 100 doesn't work.
This one?
Yeah, it has fungus inside.
Let me try first though. Maybe it works. Okay. No worse. If not working well, I can change. Okay. Alright. So how we do it? 10x. Iris diagram at 10. Make sure. That's all the way all the top. Go up, 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 up to the place you cannot touch. Okay, then go down a little bit, go down a little bit. You'll see a lot of things there. See it? Lots. See it? Again, 3 representatives from the whole classroom. That's what 10x looks like. See it? Okay, come here to see it. What it looks like? Good. Yeah, but not very much detail. You do see a big chunk of bacteria there. Okay, what we do? Let's try whether 100x is working. If it's not working, it's okay. 1 twisted, 2 twisted, you have an angle there. So we're going to add one big drop of the oil. That's called oil immersion. So we learned about that. Remove the air gap. We put a big one. We'll see whether it works. Maybe it's not. Maybe it's yes. Oh, maybe it works. Oh, it works. Good. Oh, it looks good. Okay. But the thing is for me, it looks like it's bacillus. It's not E. coli because it's very long. We'll see. Come here to see. That's what the under 100x with oil immersion, you can see. That's a very long rod. See it? Okay. See it again. That's what on this 100x looks like. What? Come to see it. Double check. That looks good. Lots, pink, saffroning, and it's long. And you guys can feeling about there's a 2 dots on the end. That could be the endospore. But this is not the endospore stain, so you're not going to see it. But I suspicious this is bacillus. But it's okay, you can do the simple stain. That's more obvious because E. coli is a short rod. Sometimes you doesn't see very clear. It's a long rod.
They might have a contamination.
They may have a contamination. So we immediately prepare staphylococcus in our lab. Might be. Maybe better for next week for the gram stain. Okay, we'll see. So that's what we do. Now, when we finished, of course, this works. So just a step down and you can clean it. Put on your own. You have a box right here. You don't have to throw them away. You can clean it. Put on your box right there. Okay. Then next time when you gram stain, you can use it. You don't have to throw them away because we have a limited budget for all of these things. Used to be you can just throw it away, but you can keep it. Okay. So I left this one as my demo. So today's work is very simple. Everybody grab one piece of glass slides, Share the plates and put the colony there. We do the simple stain. I hope you're gonna do the same like I did. It's very short, 5 minutes you get it done. Okay, very good. Use your cell phone, take the picture. You can get a big good picture.